Hello friends, welcome to BMH Learning. This video is about peroxidase activity assay. Peroxidase is an enzyme found in a wide variety of organisms, from plants to humans to bacteria. Its function is to break down hydrogen peroxide, which is one of the toxins produced as a byproduct of using oxygen for respiration. The enzyme basically catalyzes the decomposition of hydrogen peroxide into water and molecular oxygen. So these are the enzymes that catalyze oxidation reduction reaction by mechanism of free radical that transform several compounds into oxidized or polymerized products. Requirements: we are gonna need 3 chemicals: a buffer with pH 7 to 7.4, a substrate guaiacol. Third chemical we need is hydrogen peroxide. Apart from these, we need the plant extract. Procedure: let's say we are gonna make 6 samples from one source. That is, only one type of plant or vegetable we are using. First step is blanching, in which we boil the plant for a brief period and then plunge into ice water. So we are gonna blanch the plant for 5 different time frames. That implies blanching for 1 minute, 2 minutes, 3 minutes, 4 minutes and 5 minutes. That's why we are using 6 samples. This means the first sample is not blanched. Be careful not to increase blanching time, that is, if second sample is treated for 1 minute, it must be only 1 minute and we should not extend it. And this applies to all samples. To do so, we use a water bath which is set at 80 degrees centigrade. Let's suppose we do it for 1 minute sample. So, we put the sample in hot water bath set at 80 degrees centigrade and immediately start the timer and treat the sample for exactly 1 minute. The sample is then put in ice water or placed under cold running water. Similarly, we repeat the blanching process for remaining 4 time spots. Next is the preparation of sample extract. First, measure 10 ml distilled water in a measuring cylinder. Add the measured amount of distilled water into the juice container of the grinder or blender. Into this container, put 0-minute sample. Grind it. Repeat it for remaining 5 samples. Make sure one thing here, that you grind exactly same amount of plant in all samples. Let's say 50 gram in each of 6 samples. All 6 sample solutions are filtered using a filter paper. Now let's test for peroxidase activity. Take 6 test tubes and label them as 0 minute, 1 minute, 2 minutes, 3 minutes, 4 minutes and 5 minutes. Add 4.7 ml of buffer in all 6 test tubes. Next, add 0.1 ml of substrate in each test tube. Next thing we have to add is 1 ml of each different extract into its respective test tube. That is, 1 ml of 0-minute extract is added in 0-minute tube, and this is repeated for rest of the tubes. Into all these tubes, we add 0.2 ml of hydrogen peroxide. Apart from these tubes, also set one more test tube labeled as blank. Into this tube, we don't add any plant extract, and we add 5.7 ml of buffer instead of 4.7. Other chemicals are added in similar volumes as used for other samples. Once all the chemicals are added, the samples are vortexed for proper mixing of the contents. Stand for few minutes. You would observe the dark brown color in 0-minute sample and the intensity of color decrease as the blanching time is increased. So the 5-minute test tube has the most light color of all. Take the absorbance at 470 nanometer. This was all. Thanks for watching.